Imagine a future where diagnosing disease is quick and precise as typing into ChatGPT. DNA biosensors, programmable molecules made from DNA or RNA, make this possible by detecting specific sequences, acting like molecular switches. They are powerful tools used for disease diagnostics, environmental sensing, and synthetic biology. Hi, my name is Shtitha Shohardo, and today I'll share my work on designing smarter DNA biosensors using transformer-based large language models. Toehold switches are engineered RNA sequences that regulate gene expression. Each one contains a hidden ribosome binding site, or RBS, and start codon, folded into a hairpin structure that blocks translation. At the 5' end, it is exposed Toehold sequence, complementary to a specific trigger RNA. When the trigger binds, it unfolds the hairpin, revealing the RBS and allowing translation of a reporter gene like GFP. This produces a visible signal, indicating the presence of a target. These switches can be easily programmed to detect RNA from viruses like Zika, Ebola, or SARS-CoV-2. However, designing them can be difficult. Performance depends heavily on sequence-dependent RNA folding and stability. Additional tools like NUPEC and Vienna RNA use thermodynamic models to simulate RNA structure, but they often miss key biological subcomplexities. This makes Toehold design a trial and error process that's costly and slow. My research takes a different approach, using transformer-based machine learning models to learn directly from experimental data. Like ChatGPT learns language, these models learn biological patterns from sequence data, capturing complex dependencies that guide biosensor function. I used a dataset of over 990,000 Toehold sequences with experimental performance scores from a major study. I trained machine learning models to predict performance from sequence alone. I started with convolutional neural network, or CNN, which detects local patterns well, but struggles with long-range interactions. I then implemented a transformer model, which uses self-attention to capture global relationships within sequences. Transformer outperformed both the CNN and the original studies model, achieving a lower mean absolute error, demonstrating stronger predictive accuracy. I used visualization tools like Synapse and Vine via Captown to identify which parts of the sequence influence the model's predictions. This early stage work, limited to 30 training epochs and focused on prediction, sets the stage for developing a generative model to design optimized biosensor sequences from scratch. Like Storm. No trial and error necessary, just rapid, intelligent, AI-driven design. Thank you.